Okay, we're going to start. Hello, everyone, and welcome to today's AAPS eChalkTalk. Today's eChalkTalk is hosted by dsm-firmenich. You may submit questions at any time by entering them into the Q&A box of the Zoom platform. All questions will be answered at the end of the program. It is now my pleasure to introduce our moderator, Fernanda Onofre. Fernanda, the floor is yours.
Thank you very much, Stacy. And thanks everybody for joining, and thank you for the opportunity to be talking about this very interesting topic of bitterness today. So before I hand it over to our experts, I just want to give a brief description of the session and what the audience can expect to learn in this half an hour. So dsm-firmenich is an innovative leader in the supply of high quality vitamins and also taste solutions. And we're going to outline how flavors are designed, and we're going to talk a little bit more in depth about novel approaches to address bitterness in drug product formulations. So the audience will better understand the science of flavors today. You're going to understand how the flavors can modulate, can change the taste perception, how it can help overcome the taste challenges. And then we're going to go more in depth on a very interesting technology that allows us to synthesize very specific beta blockers that can be targeted at active pharmaceutical ingredients. So let me introduce you to our experts. Today we have Lisandra and Guy. Lisandra is our Global Taste Technical Director. She's got more than 20 years of experience within the flavors industry. She's got a master's in this field, and she leads various teams from applications, flavor creations in various segments. And we also have Guy. He's the head of receptor-based discovery R&D at dsm-firmenich. He's trained in receptor pharmacology in Canada. He's got a postdoc from California in molecular pharmacology and signaling. And he joined the company a few years back to head this reception-based discovery group. He's been working on functionalizing taste receptors and synthesizing all kinds of cool things that he's going to talk to you about. So with that, I'll hand it over to our experts. Lisandra, please.
Thank you, Fernanda. Thank you. Good. So let's talk about taste and flavors today and how taste plays a key role in patient compliance. So first, consumers expect products that delivers the best sensorial experience. If a medicine tastes bad, patients, especially children, are less likely to take it as prescribed. So our challenge today is really to overcome the bitterness, as more than 60% of APIs in the market are considered bitter. Therefore, I would like to explain the different approaches to mask bitterness from our human taste receptors understanding to the final design of performing masking flavor solutions. But basically, what is a flavor? So a flavor is a product that is obtained from a complex blend of can be natural or synthetic materials that once together applying a medicine or food can deliver desired taste. But concretely, how does a bitter masker flavor look like? Basically, to design the most efficient bitter masker, we need to work into a holistic approach, first by utilizing or combining one or more several basic tastes. So basic tastes are ingredients that do not have necessarily a smell, but has a taste, such sweeteners, acids, salts. And at the same time, depending on the final application, the flavor must contain also the appropriate solvent system, propylene glycol if we're talking about liquid flavors, or system like maltodextrins if in powder format. The flavors are created basically by experts who are called the flavorists, and to continue in the journey, they also going to looking at mid-notes. Mid-notes are also flavor ingredients that could either mask or block taste receptors, and could also play an important role, those ingredients, in terms of also texture, temperature, and playing with the trigeminal sensation. Trigeminal sensations, we're telling here about ingredients that can bring in cooliness, spiciness, repudiant and C, in order really to distract the bitter perception coming from those APIs. Last but not least, taupe notes, or what we call as well aroma, where are very volatile molecules that are added into the flavor composition to provide the desired smell and taste impact to support, of course, overcoming the bitterness. And here could be citrus, vanilla, and berries will perform well. So in a nutshell, basically masking bitterness, we need really to tackle through an holistic approach. So understanding, of course, the product format and the medicine formulation in order really to adapt to the masking strategy. So either liquids or powder formats or even encapsulating the maskers are necessary to prevent those maskers of interacting with the APIs and having a better stability in the final product. At the same time, while selecting the best flavoring substances, maskers, blockers, and any other ingredients, we also need to comply with the different regulatory bodies and different also consumer needs, depending on the final application, the country, and consumer groups, if it's for adults, for instance, or pediatrics formulation. As a conclusion, bitter masker solutions require very good understanding of taste receptors, and we need to have the right ingredients to play with, as well as a good understanding of different regulations. So in order to continue that explanation and going a bit more in talking about taste receptor discovery, I'm hand over here now to Guy Servan for the next part. Thank you.
Thank you, Lisandra. This is Guy here. So as Lisandra was saying, with respect to bitter taste modulation, dsm-firmenich uses mainly three approaches, and very often in the final solution provided by the company, all of these different solutions depicted here or ingredients will be combined together in the final formulation. So we're talking about bitter blocking or antagonist of the receptors, which is going to be the focus of the talk today. There's going to be some probably masking ingredients there. These are molecules that usually maybe encapsulate the bitter blocker, the bitter API or the bitter molecule, or somehow prevents the interaction of the molecule with receptors by yet unknown mechanisms. And also molecules that interfere with the taste of bitterness. You can use sweeteners, for example, to distract the off taste of certain APIs and other molecules. So very often our final solution will contain one, two, or three of these different approaches. So now deeper dive into the physiology of taste. So we detect sapid molecules that are diluted in our saliva with taste receptors that are expressed at the surface of taste receptor cells that are bundled up in these taste buds, this onion-shaped structure that you can see here on the left. And each of these taste buds contain 50 to 100 cells. And these taste buds are, as you know, located in taste papilla in the back, the side, or the front of the tongue. And we also have some taste papilla on our soft palate. We now know through work done mainly in academia that the way taste is coded at the periphery is that each taste receptor cell here expresses mainly one type of taste receptor. For example, cells that detect bitterness will not express the sweet receptor and so on. So this is how taste is coded at the periphery. And there are now 29 known receptors or ion channels that are known to be receptors for sapid molecules, and they are described in this slide. So we have on the left here, the sweet taste receptor, which is made up of two different proteins, two different GPCRs that need to assemble together to form a functional subunit. So we have here the sweet receptor, the umami receptor, a close cousin of the sweet taste receptor. They share one subunit. With respect to bitter, there are 25 bitter taste receptors in humans. And I'll go through this in a moment. These are also G protein coupled receptors. OTOP1 is a proton selective channel that is known now to be responsible, at least in part, for the detection of sour or acids. And with respect to saltiness, at least in rodents, at least in mice and rats, ENaC, the sodium channel, epithelium sodium channel, is known to be involved, at least in part, in the detection of saltiness. In humans, it's not yet so clear what is the active receptor for detection of salt. So it's an active area of research. So the way we develop new ingredients at dsm-firmenich is by the use of a classical small molecule discovery paradigm that you would find in biotech or pharma companies. So we take the genes that code for taste receptors, we express them into cells that can be kept indefinitely in the lab, and we use high-density cell plate formats to seed these cells. And on one end, on the other end, we have this proprietary collection of natural compounds or synthetic compounds that are also presented in these high-density plate formats. And the cell plate and the compound plates are presented in a sophisticated fluorescence imaging plate reader, where the machine basically dumps the compound onto the cells. And we're looking for either receptor activation, receptor inhibition, or modulation of some sort. And this way we can pick up hits. And our chemists look at these, and this is where we start the typical hit-to-lead optimization cycle, when we improve the properties of these molecules, making them more potent, more stable, more soluble, and so on over several months. And this is guided through taste tests and assay. And after several months of doing this, usually we have a candidate that goes into development. So these are the 25 bitter taste receptors that we have in humans. They're located in 3 different chromosomes. And we now know from genetic analysis from work done at our company and several other labs around the world that our sensitivity to bitter compounds is mainly driven by the genetics. So mutations that are present in these receptors will dictate whether or not we are more or less sensitive to different bitter compounds. And what are we trying to achieve here? Just to illustrate a bit more our approach is, if you look here on the left panel, we have a receptor here, this cartoon of the 7 transmembrane domain of a GPCR that is embedded in membrane. The top would be the outside of the cell, the bottom would be the inside of the cell, and then you have a bitter ligand, or an API in that case, binding to its pocket, and then that leads to an activated receptor and ultimately a bitter taste when the signal is transmitted to the brain. What we're trying to do here is to develop molecules that will either do one of 2 things: either block the bitter ligands from binding to the receptor, that's one approach, or bind to the receptor and keep it in an inactive state, right? So in a way that when the API comes, for example, it cannot generate a signal and a bitter taste. So this slide here represents some of the work that is published, some of the work we've done with our first venture into this field years ago. What we're looking at here on the top left panel are raw data from a screening plate, 384 wells. The far left columns here are just cells that are stimulated with buffer. Little or no fluorescent signal is generated. On the far right here, you have columns that are stimulated with a suboptimal concentration of a bitter agonist. It could be an API, for example, where we use a concentration that elicits about 80% receptor activity. And from this point to that point here, this is the assay window, right? The dynamic range of the assay. And in the middle here, you have the same API or bitter compound that is mixed with compound libraries that could be either activators or inhibitors of the receptor. And using very simple stats there, we determine what is a cutoff for primary hits. And you can see here, for example, 2 wells or that would contain a compound that is potentially very interesting antagonist for that receptor. And you go through this with thousands of plates, right? And then you look at all the activity in a bin distribution here, and you look at the primary hits, and this is where we pick these, and we go through dose response analysis and other pharmacological analysis. And ultimately, after several months of work, you generate a molecule that is a very good antagonist of this receptor with sub-micromolar potency. And very importantly, this program was initially a collaboration we had with Nestle. They wanted a bitter taste receptor antagonist for coffee, and we were able to deliver on this. This is a taste results with a sensory panel showing that the sample that contains the antagonist is much less bitter than the sample that does not contain the antagonist. So now, if we can do this for coffee or any other bitter taste ingredients, we can do this for APIs. As you learned from Lisandra, most APIs, certainly more than half of the APIs, are known to have a bitter taste, and at least 25% of them are characterized as very, very bitter. So, and this is what we did. More recently, we developed identified and developed a very, very potent bitter taste receptor antagonist for known APIs contained in very popular OTC formulations. So this is some of the data here showing that this antagonist can block the receptor activated by acetaminophen with, again, nanomolar potency, right? In panelists, in taste data with panelists, we can see that concentrations as low as 10 ppm are enough to attenuate the bitter taste of acetaminophen, as well as guaifenesin here at 50 ppm. We also have some data with dextromethorphan showing this is also very promising. So again, we have this molecule is a very effective and efficient bitter taste blocker of known APIs. And again, we can do this on several APIs. It doesn't have to be these OTC APIs. It can be any API of your choosing. So we hoped that you've been able to understand today that the majority of APIs exhibit a bitter off taste, that existing solutions, whether they're sweetening or masking, are not sufficient. I mean, everybody that has a kid, for example, that has tried yesterday to provide antibiotic would realize how difficult this is, or even over-the-counter cold medication. They really don't like it, right? Clearly, even the current solutions without bitter taste blockers are not sufficient. So we feel that one ingredient we can add to our toolbox are bitter taste receptors antagonists or blockers, and really they show promise. And I think one very important aspect of dsm-firmenich, what we can offer here, is really an end-to-end process where we don't have only the receptor assays, but we have the flavorists, we have the overall ensemble of knowledge that allows us to deliver the best solution. So with that, I think Fernanda will take questions.
Absolutely. Thank you, Guy, and thank you, Lisandra. We have about 10 minutes for questions. There's a few questions that have come up on the chat. Please feel free to continue to type your questions in, and I'll take them. I think there is a couple of them that have been answered, sorry, but I'll ask them anyway just to make sure that the audience is clear. So, Guy, those are for you. How do you screen taste masking agents without giving to humans? Are there any in vitro models or animal models to evaluate formulations with taste masking agents for formulation with bitter API? I think you've touched a lot on that, but if you can just reiterate.
I'm not sure what you should understand the question. So with the bitter API, I mean, the API itself can be used in the assay to show that we can block the API activity on the receptor. Now, when it comes, is the question related to how can we evaluate the API in taste test?
It's how we can evaluate the bitterness without giving it to humans.
Oh, well, what we've known over the years, we've done this for 25 years now. We've built a huge database and knowledge of activation profile of bitter receptors in vitro and the correlating efficacy or effect in vivo, right? So we know that whenever we can activate a bitter taste receptor, we find the dominant bitter taste receptor, and we see an EC50 of a certain value. We know that if we go 2 to 3 to 5-fold above that EC50 in human taste test, it's going to be bitter tasting. So I think, the assays that we have are very, very predictable of the taste outcome. So I can guarantee you that if you have an API that activates 1, 2, or 3 of these receptors, it's bitter. And depending on the level of activity and the dose at which it activates, we can probably even determine how bitter it's going to be. So, does that answer the question?
I believe so. Thank you. And you touched on a point on a second question here. How likely is it that a bitter drug will activate only one bitter receptor? Mm-hmm. And the second question within this question is, how many bitter blockers are commercially available and how many receptors do these block?
Okay. For the first question, that really that comes to the core of what we're trying to do here. Yeah, the APIs, most of the time, an API will activate 1 to 5 bitter taste receptors, right? It's very seldom that we are going to find an API that only activates one receptor. Acetaminophen is an exception, only activates one receptor, for example, but most other APIs activate several bitter taste receptors. What we found out, however, there is always a dominant receptor that is either activated at much lower concentrations of the API or that elicit much greater response of the receptor. So what we learned over the years is that if we target our discovery on the dominant receptor and develop antagonists, we can significantly attenuate bitterness in products. So now, can we fully block bitterness? Most often, no, because again, as the question implies, more than one receptor are activated. But if one targets the dominant receptor, one can have a significant effect on the outcome. And then ultimately, the remaining of the bitterness note can be even further attenuated with maskers, sweeteners, and other approaches. So it's a great way of approaching the problem. Now, how many bitter taste receptor antagonists are on the market? We have commercialized one so far. This is the one I've presented that blocks the bitter taste of coffee. We know that it blocks the bitter taste of other compounds, including APIs. So clearly, I mean, if we develop a bitter taste blocker for a receptor that is activated by coffee, it turns out that APIs also activate the same receptor, then obviously we may have a solution. So, the molecule we use to activate the receptor doesn't really matter. And we have one commercialized and we have one in development, the one I showed at the end.
Perfect. Thank you. An interesting one for you, Guy. Are the bitter receptors homologous across species? For example, dogs, cats, how is it similar or different?
They're very, very different. Between rodents and humans, there's some clear orthologs. But I don't recall exactly by heart the percent homology. But it's very often difficult to translate activation profile of bitter receptors in rodents, for example, to the corresponding receptor in humans. So it's been done for a handful of them, but I would say for the vast majority, it's not a clear correlation. There are not clear orthologs between human and mice. I don't know. I would not recall about other species, but I would imagine it's the same story.
Okay. So there is a couple of questions around the regulatory aspects of these molecules as we are in a pharma setting. So we were definitely expecting those. There is a couple of similar questions here. What is the regulatory pathway for this new bitter taste receptor antagonist? What are the safety studies done? How could this be used in a pediatric formulation, for example, that requires a lot of safety demonstrations of its ingredients? So, Guy, can you just, at least from your point of view and with the molecule in its infant stage, how does the regulatory pathway look like? And then, of course, we have the regulatory pathway for the drug product, which is another topic. But from the compound itself, once it gets out of your lab, usually, what do we do?
Yeah, and this is published information. We can certainly send some of the papers we published over the years on how these flavor modifiers go through regulatory approval and what type of safety data we provide. we provide. They go to very rigorous assessment of genotoxicity and toxicology in animal models, very similar to what one would find for a drug. And we do this usually up to now. We've done this to get FEMA approval or EFSA, which are the regulatory approval bodies around the world for flavors and flavor modifiers. Right now, I think with the help of Fernanda and her team, we're looking into how to go into the excipient world with some of these molecules, which will probably require even additional safety studies. Up to now, we do even for FEMA GRAS for flavor to be able to use these compounds as flavors, they go to rigorous assessment of safety. And again, this data is published certainly for BB68, what I just showed, and others.
Yeah, and we use the if I can just compliment you, we use the extensive safety testing that we do as a basis to support our customers when they are using this ingredient as part of a flavor formulation in a drug product. Because to Lisandra's point earlier, this would not be used as a sole molecule. This is part of a solution which may contain several ingredients, right? So the bitter blocker is one. So there is a robust safety package that comes with that ingredient within the flavor formulation, right? And so that is what we can provide to our customers to support their submission to the authorities to answer questions around the safety of use. And of course, we are here to help our customers through the regulatory journey of submitting a drug product containing a flavor, whether it's pediatric or for adults. Right? We have a lot of questions coming in now. Let me go through this one. Can we use the plate system, Guy? I think that's for you, to assess, it's written here, the flavor profile, but I guess the bitterness profile of a new chemical entity, a new API that has never been tasted before. If yes, could this be offered as a service, or, can we do this? So.
Absolutely. I mean, we've done this in the past, and then Fernanda, actually, you want to comment on this? We're currently trying to carve out a new program where we can offer this type of service.
Yeah. So absolutely, this is an interesting approach to really assess those molecules where we're just in discovery stage, or that we understand that it's going to be administered through a route where, taste is going to be a challenge. So we can definitely use a lot of the in vitro work that Guy and his team can do in our San Diego facilities to really understand, especially the bitterness, which seems to be a prevalent problem in pharma, right? Of these chemical entities. So definitely. Have you found that certain active compounds have a broad spectrum of application to blocking bitter taste? In other words, is there one blocker that works for many different types of bitter tasting drugs, in your opinion?
Bitter tasting drugs, I mean, that I don't know. I mean, I would presume yes. The reason why I'm saying this is that when we profile bitter receptor antagonists, either the ones that are commercialized or the ones that are in the pipeline, we have more than one, the one we've showed you here. And when we look at their capability to block the bitterness of different ingredients, different either from plants or from different, food or beverages. Very often we realize that an antagonist developed for one thing turns out to be very active for a lot of different bitter molecules. Now, the reason why I cannot specifically say for APIs is because we've just started more recently to be able to taste test APIs in a more routine way, or maybe routine is even taking it to the next level. Tasting APIs at our company is under much more greater scrutiny. So we cannot yet, we haven't done yet a lot of tasting studies, with a broad range of APIs. But I would imagine from what we've learned with bitter compounds in general, that the same story would apply.
Thank you. Lisandra, quick question for you. How many ingredients on average are there in a flavor formulation?
Well, good question, Fernanda. I would say there is as many as we expect to create a desired taste, but in average, of course, the idea is not to complexify operations and the way we create those bitter solutions. For example, bitter masker flavors, in average between 15 to 20 ingredients. Today we are able, of course, to create the larger spectrum of either blocking, either modulating, and at the same time providing the desirable taste. So in generally speaking, 15 is a good average, I would say.
Thank you. And Guy, if you can go to the next slide, there is our contact information there. That one. I think we have time for, Guy, there's one question. There's two small questions within one for you. Do these antagonists have a taste of their own? And then is the blocking effect reversible?
Yes, that's a very good question. So yeah, the taste on their own. Sometimes as we develop these molecules, they have, and then we have to work around these and optimize them. For example, some bitter antagonists initially, as we develop and we taste them, we realize, oh, they have bitter taste on their own. They're small molecules, right? So we have to de-optimize the bitter tastes, or they're also agonists, to de-optimize the agonist property while keeping the antagonist property. But to make a long story short, once something goes into development, the goal is that they have no inherent taste of their own, right? So that's the first thing. The second thing is regarding reversibility. Yeah, once you swallowed whatever's in your mouth and the saliva dilutes the environment at proximity to the receptors, the antagonist will come off, right? So yeah, it's totally reversible. And there's typically no long-lasting effect of bitterness blocking. So if you take your sample and then you swallow, and 10 seconds later you taste again the same bitter compound, now it's going to be highly bitter because the antagonist is not there anymore.
Excellent, thank you. So I think, Stacy, we're coming to the end of our slot. So there are a few more questions in the Q&A. We are going to get those questions. We can address them with the audience directly. Please leave your contact information as well. So I want to thank Guy and Lisandra for sharing your knowledge. Very interesting presentation. I really appreciate your time. I hope the audience learned a lot today about bitterness and bitter blockers, and hope we can be of help in your future developments. So thank you very much. Thank you. Thank you, Stacy.
Thank you.
Thank you.